Olá, bom dia a todos. Então, obrigada mais uma vez pela oportunidade de trazer um convidado da oftalmologia, que tem que ter uma interseção com a reumatologia, mas é sempre uma oportunidade muito grande também para os oftalmologistas terem aqui também algum palco e trazerem alguém que eu espero que seja inspirador e que também possa eventualmente também fomentar novas linhas de investigação. A Lola Solebo, só por uma apresentação muito rápida, é uma Welcome Principal Clinical Research Fellow na UCL e é oftalmologista pediátrica muito dedicada às uveites, embora também faça oftalmologia pediátrica per si no Great Ormond Street Hospital, e é líder do CHIRE, que é o Childhood Ocular Inflammatory Research Research Group, que tem como objetivo a investigação do ponto de vista epidemiológico e de data science nas uveites pediátricas. E por isso eu vou-lhe dar a palavra, Lola.
Thank you so much. Shall I use both?
Bom dia, estimados colegas. Did I say that okay? Thank you so much for the invitation here. So, first of all, I need to announce that I am a huge fraud. My first fellowship was in straightforward pediatric ophthalmology, so all of pediatric ophthalmology, because my PhD had been in congenital cataract, one of the most significant causes of childhood blindness globally. And when I had an interest in congenital cataract, it was around the time when we were starting to use artificial intraocular lenses in very, very young children, in infants and in babies. And we designed a study, a national cohort study that identified children who were undergoing surgery with or without this artificial intraocular lens to try and uncover, using an observational approach, whether or not intraocular lens were indeed related to better outcomes. And I started the cohort off doing PhD, had a few babies, then came back as a clinical lecturer to do the 5-year follow-up. I know this is not exactly the target audience. I'm going to describe what the impacts were on practice and on policy, and then swing back to talk about methods, because it was really that scientific, rigorous training that that's what the PhD is for, less the clinical area of expertise, more the scientific skills you are picking up. And I am an epidemiologist. I am a counter. So the headline findings from that study of congenital cataract were that primary intraocular lens implantation, routine for adults, was not related to better visual outcomes for children with congenital or infantile cataract. In fact, for those children, it gave them a greater than 6 odds chance of needing subsequent surgery during that first year after their first surgery. And that repeated surgery is, of course, happening at a really critical developmental time for that very, very young brain. So those findings led to lots of papers and some prizes, but really pleasingly, what they led to, as well as that change in clinical practice, was a change in not just national, but also supranational health policy. I was involved with the World Health Organization's development of the package of eye care interventions for children globally. But as I say, it was a method. It was a method. So for IOL under 2, which is what the cataract study was called, the first thing was understanding that you wanted an interception cohort. That is, you wanted prospective data collection from the time when the first interesting exposure happened in that individual over time, so that longitudinal approach. For this study, we also used active surveillance, and active surveillance being that epidemiological term that means that you're creating a network of involved clinicians, you're asking them to let you know when they see a case, but you're also asking them to confirm the absence of a case. We had a standardized data set. So in epidemiology, the data collection form, that is your test tube, that is your experiment. So you're ensuring that you collect the right data and harmonized definitions. So at least a year spent across the team agreeing that we were all speaking the same language, and then implementing the methods across a multi-center group. And of course, it's an observational study. You're seeking to derive causal relationships from that messy data. It's really important to upskill yourself with understanding how to deal with that clustered data. And these are all the skill sets that we then put forwards into Unicorns, which is the national, so it's multi-center across the UK, national uveitis and childhood prospective cohort study. The only thing that was missing was the active surveillance. And Unicorn started in March 2020. There were many other things happening for the first time in March 2020 across the world. So active surveillance was one of the things that we knew that we wouldn't necessarily get absolutely 100% ascertainment across the country as the pandemic was roaring away. I will get it right one day. But if I start with what epidemiology is, and epidemiology is understanding burden, but also understanding etiology, counting who gets what, so you can understand the why. The harmonized data set was an essential part of designing the experiment that was Unicorns. So first of all, sitting down and understanding who across the country was looking after these children, and then getting them to do that harmonization of terminology. So what do you mean by the different clinical terms you use? What do you mean by the different phenotypes that you're using? And then developing a core national data set, really time consuming, but essential for the work that we planned for Unicorns, but also essential for the future work that we plan. We want to make sure that doing something like Unicorns or like IL-2, that is designing that observational study where you have field work and you travel around transcribing from medical records, we want to make sure that that kind of science is obsolete, i.e. pulling research-ready data sets out of electronic health care records. And the sort of credentials that we've put together and the clinical network that we put together nationally means that now that we're translating the clinical data set into SNOMED CT, which is a nomenclature used for clinical terminology globally, so that we can go into electronic health records. For now, we're working with something called EPIC and also Medicite. And we're working not just within the UK, but partnering with other nations who use these electronic care records to try and pull out research-ready data sets out of the messy observational clinical data so we can dig into research for this rare disease. To stand here. So Unicorns, although we started in 2020, between 2020 and 2023 is when we looked at the inception, designed the inception cohort to identify new episodes of childhood uveitis. So although now we're at the 5-year stage, we haven't yet started to collect the 5-year outcome data. But what I can talk about is what we understand about the incidence of uveitis, despite the fact that we were stopped by the pandemic from having a true rigorous active surveillance approach. And what we understand with regards to the incidence of uveitis is, unlike the evidence base that already exists in the UK, through this rigorous multi-centre approach, we're able to declare that at the time of diagnosis, only 15% of those children have juvenile idiopathic arthritis. So in the textbooks, the prevalence of JIA amongst children with uveitis runs from between 30% to 90%, depending on which paper you're reading. But in the UK, from this rigorous approach, looking at all the nation, we found that JIA was really underrepresented, and that has various consequences. Number one, all our mindset around picking up uveitis early enough to make a difference to that clinical trajectory is screening. You have a population that you know are at risk. If you watch them closely enough, you can pick up the new cases to intervene early enough to prevent the complications from occurring. However, if the majority of the cases are happening outside of the known population at risk, it means that we need to examine the primary care routes, the emergency care routes into getting care for children with uveitis. We also looked at the ethnicity and the socio-economic background of the children with uveitis, and we found that it was overrepresented. So with regards to what the ethnicity was, relative incidence was higher amongst children from an Asian British background and Black British backgrounds in particular. With regards to the deprivation score, so this is with your postcode at which the child lives, how do they rank nationally with regards to the deprivation around their area for things like health, environment, education, crime outcomes. And we found again that if children lived in the most deprived areas in the UK, they were much, much more likely to be diagnosed with uveitis. So again, lots of interesting hypothesis generating findings that we have that we can use to craft and design the further observational studies that we undertake through Unicorns. the other thing that we found, which I think comes up really well, is that the age of diagnosis in years, although we knew before that uveitis had a bimodal distribution with a peak in early childhood, predominantly JIA, and another peak later on, the existing evidence base suggested that the primary peak was in the early years with a smaller hump later on. But from unicorns, we identified that actually that primary peak is happening in adolescence, during that hormonal storm of adolescence. Of course, this is the relative picture of uveitis, and one of two things, or both, could be true. Either the JIA cases are reducing over time compared to previous literature, or the non-JIA cases are going up over time compared to literature, and both may be true. And indeed, other investigators have identified a reducing incidence of JIA uveitis because of the speed with which we now intervene with DMARDs and with biologics in children with JIA. But it does mean, of course, that with this relative change in how disease is patterning across the nation, we need to re-understand what we need to do to provide care to those children, but also re-understand and provide focus to those children who do not have JIA at the time they are diagnosed, and indeed never go on to develop JIA later on in their life. Now moving on to everybody counts. So health equity and understanding what equity really means, i.e. you give more support to the people who are more in need, is a burning issue globally now, and rightfully so. We understand that some children, some families need more. And in the UK, if you're a child from an ethnic minority background, or you're a child living in poverty, you're more likely to have an eye disorders. Almost all the eye disorders more likely to occur in poorer children or children from ethnic minority backgrounds. If you do have an eye disease, you're more likely to have a more severe manifestation of that eye disease. You're less likely to undergo innovative treatments. If you look to see who's being offered the new jazzy treatments, it's more likely going to those children who are from richer backgrounds or from majority ethnicity backgrounds. And you're also more likely to have worse outcomes from the treatment. You're also more likely to have other vulnerabilities that impact on your treatment. So these families are the ones who have the most to gain from the health interventions, but also the most to lose. And much of that work comes from the work we did on cataract, the work we did on vision impairment in the UK, but of course now compounding the message with the signals that we're finding with unicorns, with childhood ocular inflammation also having a socioeconomic patterning to it. One of the really interesting findings from the congenital cataract study. So I remember when my interest in uveitis was first switched on, and it was when I was working on congenital cataract work. So cataract surgery is in of itself an inflammogenic procedure. Once you release the lens fibers inside the eye, you'll kick off an intense inflammatory. Children with uveitis quiet before they have the cataract surgery. So everyone gets inflammation after cataract surgery. In this national study of children who were majority aged under 6 months at surgery, the strongest predictor of how severe the inflammation was after surgery was where they lived, whether they lived in an area of high deprivation. That was driving the inflammation. That wasn't an expectation. That wasn't something we were planning to find. We hadn't collected information on deeper socioeconomic information, so it was just really enticing to me, and that's one of the things that first got me into inflammation as a whole body disease, and actually not just a whole body disease, but a whole sort of society disease in which you would get interesting heterogeneous phenotypes. You get a lot of quality of life information because of the burden of disease. You are dealing with socioeconomic factors as well, and also, of course, the chronicity of disease clinically means you build up such a close relationship to these families, such a precious close relationship to these families, unlike with cataract when you operate and you sort of let them go over time afterwards. Sometimes it works, sometimes it doesn't. As well as that inclusive approach with regards to making sure you understand how the socioeconomic drivers can be influencing disease, we also wanted to make sure that we did something different with uveitis with regards to including the voices of the affected children. As I showed you before with that socioeconomic patterning, we know that there's burden on burden on burden. These families have a really heavy load, and helping them to articulate what the disease means to them has been enlightening, but it's also influenced the science as well, which I'll show you later. So these are some pictures that some children drew when we asked them what uveitis means to them. So this is, I'm ashamed to say that the 12-year-old with sarcoidosis had to explain this to me. I didn't see it first of all, but the face of the person walking by is formed by the posterior synechia, so the iris being adherent to the lens. That's what she drew. This was drawn by a child who never talks to me in clinic because they're there's a lot going on. But when she, after drawing that, it led to some really lovely conversations about how the disease had affected her. And this was drawn by a child who's got heterochromia of the iris because of previous episodes of uveitis. So another way, so a cool way to communicate with these children, but it's also been incredibly influential to the science. So we created an expert study steering group, which is made of 3 parents who've looked after children who are parents of children with the disease, and also 3 young people who themselves have had uveitis. And through them, they suggested certain projects that we can undertake. One was developing a childhood onset rare disease self-care support passport that we then published on because it had a positive impact. We have a YouTube channel, which again, suggesting of them, which does a couple of things. It helps to feed back the science that we do to the key stakeholders, the families, but also what it does is it's propaganda. It keeps them interested in our research and keeps that participant pool moving for our future research. And the other thing that they suggested, I'm an epidemiologist. I do not do bench science, but they were so very always asking about why does my child have uveitis? Why does my child have uveitis? That I partnered up with someone who I think you know very well, who, for those who were here a year ago, heard Lizzie Rosser, the B-cell immunologist. So I partnered with her to create this biobank called Choir 2 years ago, where we bank tears, we bank aqueous from any children undergoing intraocular surgery, we bank blood, and we also are beginning to bank stool as well. And we're banking them through a true research tissue biobank approach where scientists can apply to get access to our tissues. And it's already supported one project that's been driven by Lizzie and her PhD student, Beth as well. And from this work, which took a combination of samples from Choir, also historical samples from an adult eye hospital called Moorfields, and also some experimental animal models, it uncovered that JR uveitis, redefining JR uveitis as a B-cell driven disorder where B-T cell interactions really drove disease, and showing mechanistic evidence to support that with identification of specific pathogenic B-cell subsets, and particularly the double negative 1 subset, and also showing increased clonality in the B-cell receptor repertoire, which again supports JR uveitis being an autoimmune disease with chronic antigen stimulation. And also from the experimental model showing that if you disrupted the B-T cell interactions, it reduced uveitis severity, which of course hints tantalizingly to new therapeutic approaches for these children. Okay, so I'm going to talk about how we've been applying epidemiology and data science to uveitis in particular, but we're going to be expanding it to other inflammatory eye diseases. So this is one of my early guinea pigs, my daughter, who is now 11, sitting on a machine called an anterior segment OCT. So optical coherence tomography, it's revolutionized ophthalmology, and now we can capture images of the front of the eye. And through these images of the front of the eye, I'm not sure how well this is showing, you might have to believe me, that at the different grades of uveitis severity, as denoted by the standardization of uveitis nomenclature, which goes from 0 to 4, but here we've got 0, so inactive, 0.5, or depending on who you're speaking to, low levels of inactivity, or actually an active eye, Sun 1+, Sun 2+, so going up in different in severity, showing that if you look at anterior segment OCTs at these different levels, you will identify different densities of the hyperreflective particles that we suggest are inflammatory cells in the eye's anterior chamber. You can still, in inactive disease, declared inactive at the slit lamp by multiple uveitis specialists, you can still see these hyperreflective particles. So one thing we did, designed an experiment using healthy eyes. So 300 children primary school age, secondary school age. Let's do anterior segment OCTs on all of them and see what the normative distribution is, so we can understand better what it means when we see particles in children who are clinically inactive. And indeed, you do see these hyperreflective particles in the apparently healthy eyes of apparently healthy children. Interestingly enough, the older those children are, so this is a cross-sectional study, but not suggesting that this is a developmental trajectory, it's cross-sectional, but the older those children are, the more particles you tend to see in their eyes, meaning, of course, that we might need to take an age-stratified approach to what normal is with regards to defining inactivity or clinical remission in children with uveitis. So data science means many things to different people, obviously. So I'm just going to be really clear that when I speak about data science, I mean the overlap around domain expertise, which is epidemiology, but also, of course, is pediatric ophthalmology, maths and statistics, the way that you handle data, but also computer science, particularly software, and of course, bringing AI into the field as well. And that's that overlapping area that is important. And I think that this area, so epidemiology in particular, is often an afterthought, but as we all know, that sort of great maxim, garbage in, garbage out. If you don't get the data set right, if you don't understand where the data set comes from, then everything you do falls down. It has no robust foundation. And that's the approach that we've taken with regards to moving from discovery of how cool anterior segment OCT could be to population and patient benefit, designing the experiments that we need to do to understand whether we can analytically validate what we're finding, whether there's clinical validation data that holds up to what we need, and also using that information to get through the regulatory approvals before we get to implementation. And that's what we've done, essentially. So starting off with test, retest, stability. If you image that same eye a minute apart, what do you find? Can you be sure that you're seeing what you think you're seeing? Understanding sensitivity and specificity, of course, and predictive values, but also understanding bias. In which populations might this not work? And you know, if you think about age, of course, there's a different population, understanding how it changes over the age stratifications. And the other thing that we've been able to work towards is biological validation. You see these hyperreflective particles. What are the particles? In one way, it almost doesn't matter. If you prove through a longitudinal study that those particles are linked to worse clinical outcomes, it doesn't matter what those particles are, they stand up as a biomarker. But to win hearts and minds, what we've been doing is imaging children who have the aqueous samples taken, so we can correlate what we see on the imaging with what the biological findings are for those eyes. All this work has led to a great deal of data, drowning in data. Sometimes I go to sleep and I've been dreaming about anterior segment OCT scans that we've been looking at. And one of the cool things that we've done, if you scan that QR code, it takes you to something called Zooniverse, which is the world's biggest citizen science platform. I don't know if you know about it. It really came to attention during the pandemic when people at home who were stuck at home would go onto it and would do some planet gazing. So scientists with a lot of data, and they need that data annotating for the purpose of their science, can upload the data with the correct ethics and regulatory approval, upload that data and ask members of the public to annotate them. So we had members of the public to annotate, is it a good quality scan? And what are the artifacts that you can see that are disrupting the scan? Can you see any particles in that scan? And can you mark those particles for me? And if you create a group mind approach to analysis, so you use 7 citizen scientists for each scan, and because we had so many people volunteer, we had that data. By taking that group effect, they can push through, they can just eat their way through lots of data and get lots of annotation, which is what we need to design the machine learning and the deep learning algorithms that will help to automate the analysis of these images. The other thing, of course, that's quite cool is feeding back. Remember the YouTube channel? We always have an eye on propaganda, because what we're hoping to do at the end of this year is launch a disease registry. So we want to make sure that we have a visible platform on social media, on for ill or for good. We want to make sure that we're not just a trusted voice, so people know that they can come to us because we will give back to them, but also we're a place where interesting things happen and they know that people are interested in their disease. And the video that we created for the Zooniverse Citizen Scientist video has been watched by 19,000 people, which again is one way we're just keeping that interest going for when we keep our research going nationally. The automation aspect is interesting because actually automating the analysis of those hyperreflective dots, that you don't really need intelligence for that. You can write a program in an ImageJ. So you can write a program in a very basic image analysis platform to do those dots. The things where you need intelligence are actually quality assessment. So is it a good scan or not? So automating that process using the annotations from citizen scientists, drawing regions of interest, again, automating that. And in partnership with the lovely Boyu Chen, who's doing a PhD, supervised by myself and Paul Taylor, who directs the UCL Institute of Health Informatics, we've been working our way towards all those algorithms. If you go up to that QR code, the code we're making open source so other people can build and build and build on it, which is again another approach that we've always taken. Those pictures haven't come out well, you have to believe me that it works. And so we're working up towards the final regulatory approvals and implementation. But one thing that always strikes me is that I think it's a cool idea, and some of the families think it's a cool idea, to move between the slit lamp, where we try and count all those white particles and have poor intra and inter observer agreements. I think it's cool to move from that to the anterior segment OCT, that you can show the family and really convince about disease. But other people have thought that their ideas were cool before. I don't know if you know what this keyboard is. This keyboard is? So this is the Dvorak keyboard. We all, of course, use the QWERTY keyboard. So the QWERTY keyboard was designed in the 1870s, and it was designed to be slow. It was designed to be slow for an English speaker, because at that time with the manual typewriters, if you type too fast, you clogged up the keys. So this is designed to slow down. And this is also designed to be anti-intuitive with regards to where most romantic-based languages languages use their keys. The Dvorak keyboard is faster. There's less repetitive strain. It has the keys where you think it should be. So, we should all be using Dvorak because the evidence base suggests that that is the best way to go. But despite the evidence base strongly suggesting that we should all be using that, we still stick with the QWERTY, because as we all know, anyone who's trying to have a new innovation, there's a lot of things that drive whether or not that innovation gets implemented into the community. And that can be consumer demand, do the patients, commissioners actually want it? Is there a cost? And it's not just a cost financially, but obviously it's a cost medically. Imagine the time it would take to unlearn QWERTY and then learn the Dvorak keyboard. And of course, the magnitude of benefit. How much more would you be getting if you took on this new implementation? And again, all that is what we've taken in mind when moving to the next step. We've always made sure that we timed how long it took to take the anterior segment OCT. And not just, some people just time it from chin on press the button. We time it from bum on chair to good image acquired, because of course getting a bum on a chair for a child is not the same as getting a bum on the chair for an adult. Oh, by the way, so over time it went down. This is my 3-year-old, which shows much longer time to acquire images. It speaks to my terrible parenting skills. That was in the very early days. And the other thing we thought about, lots of the other observers, and correctly so, focus on that grid I showed you. As the sun goes up, the particles go up. As the sun goes up, the particles go up. And as it goes up, the particles go up. As it goes up. So it's lovely that we're all agreeing on that. If you get more active, you see more particles. But we need to really dig a little bit deeper with regards to how does it go across platforms? Same eye on different platforms, how does that work? The test retest I spoke to before. Can we get image outcomes beyond quantification? So this is one paper that we did that was looking not just at different platforms, but also some qualitative work, capturing the ideas and the feelings of the family themselves. So one of the children saying it's a good idea because we can actually see what is going on. People can look and see what's happening with their eyes. This kind of evidence base and having that to hand will be really powerful when we come to regulatory approval. It's not always true. One of the most striking things I remember was very insensitive, and I only really understood it deeply when my own child was ill. But I remember in the very early days when anterior segment OCT, one of the parents who'd been really really excited about the study wanted to see their child's eye, and I was really happy to show them. And they had lots of dots, and I said, look, and the dots, and then she just burst out crying. She just burst out crying because it's the first time she'd seen her baby's eye on fire. So there is sensitivity around that, but overall the community have been very much in support. And now we're at the stage where we're doing a clinical validation study. So feasibility, randomized controlled trial of anterior segment OCT versus not SLE as you understand it, but slit lamp examination for uveitis surveillance in GIA. So from the point of the children entering the screening, they are either randomized to the OCT or to slit lamp. And that is, we have 2 more weeks of recruitment, and then we're in the follow-up stage. That's funded by Fight for Sight, a UK charity for sight loss. The lovely Dr. Sonali Dave is my postdoc who's running the study, and we've chosen that picture as the logo for UV-Screen, and the QR code takes you to the protocol. But Ines has my email. If you need any literature from me, please let me know and I'll forward it. Another interesting thing I said was beyond quantification. So this is a raw image that you get on one of the higher resolution, faster OCTs called a swept source OCT. And this is made by Heidelberg called the Anterion. So this is the image that you'll see on screen. If I binarize that image and then show you the particles in greater detail, then if I also make the particles bigger, you'll see that not all the particles look the same. They're different sizes. They appear to be different morphologies. So interesting, can we dig into what we're seeing with regards to the morphology, the shape, the size, even the reflectivity of the signal from the anterior chamber, and do, in effect, a non-invasive tissue biopsy? And that, again, is something we'll be looking to see whether or not we can dig down to in future work. Another interesting thing is not just going beyond quantification, but going beyond the eye. So this is a very, very small pilot study using OCT angiography, so non-invasive mapping out of the plexus of vessels in the back of the eye, showing that for children with GIA with active disease, an N of 12, age 5 to 10, using the vascular density scores in children who do not have uveitis, who've gone through their GIA without uveitis, but have activity is denoted by the managing rheumatologist, you can see completely drop out on the OCTA. So again, we're digging into what these biomarkers might be and how useful they might be for other disorders managed under pediatric rheumatology at GOSH. And as I said, the families have been fantastic with regards to keeping us engaged, keeping us on track, keeping us honest and driving towards the outcomes that they want to see from our research. They've also been great for hypothesis generation. And with regards to that, it's going to be a really interesting time over the next 5 years because we have funding for Wellcome to grow what we're doing. So not just looking at the imaging biomarkers, but creating that rare disease registry I spoke to you about. Also digging into that electronic patient record. We know what the harmonized data sets look like. We've got agreement on what the SNOMED terminology should be for that capture. We can now go into electronic health records across different hospitals and pull out the data and start to derive research-ready data sets to dig into the determinants of disease. And also what we can do is link that to administrative health data sets across the UK to start to understand what the long-term outcomes are for these affected children. So we have Ajita, who is a clinician scientist who is responsible for much of the image analysis collection. Sonali, who you've met before, who runs the UV-Screen screening study. Maddie, who's our PPI face, who helps to really drive community choir. So we keep the parents included and the children included in everything that we do. Jin Peng, who is a fantastic computer scientist, who's working on the taking forward the automated algorithm, and also is developing a quantum OCT, so using a quantum chip to turn that big OCT machine you saw into a handheld OCT, so we can really make that modality more flexible. And also Mong Yun, who's working with our routine data collection. So for now, she's starting on a data set of 670 children with uveitis for whom we have at least 12 years of outcome, trying to understand what other healthcare utilization they've had for what other disorders emerge later on in time. So I look forward to maybe visiting you again and letting you know what we found. We've only just started that journey as of the middle of last year. And these people have only been in place since January and February of this year. So we're a very new team. Okay, so I hope I've left enough time for, oh gosh, I spoke too fast. Oh, I hope I've left enough time for questions. Yeah, oh gosh. Thank you.
Thank you. It was very interesting and were very fun and compelling, and I think everyone was really convinced about how fun managing childhood uveitis is. Not so fun, but very interesting and very rewarding as physicians, I think, as clinicians, and also from the researchers' perspective as well. So anyone would be happy to ask some questions, Felipe? Felipe is a pediatric rheumatologist. She's the head of the pediatric rheumatology, so very important person in my life.
Hi, thank you so much for your excellent talk. Given that, well, most of uveitis cases in children apparently are not GIA associated, could the current screening strategies that are so much focused on GIA now could be missing a substantial proportion of clinical significant uveitis?
And that tells us two things. One, there is enough awareness. Conjunctivitis and the other thing is that we need to take for 2 seconds. So OCTs, they're sitting in every room. High street opticians. So, yeah. Those machines, many of them are sort of it can be the awareness.
They need to confidently say, yes, something is going on, or no, you are completely well, and that should hopefully get them in to see me faster. But whole population screening for rare diseases is tricky, because if you know exactly when the disease will occur, then you can do whole population screening. So
One stopped working.
oh, okay. So you know, congenital disorders, you know that you should screen. Familial cases, you know that you should screen. But for so many of these, it comes out of a blue sky. But luckily, if we know they're getting symptoms and the symptoms are the big flag that something is happening, then we should make sure the pathway for symptomatic presentation is better. That was a very long answer. I hope that's it.
Thank you for this very elegant and enthusiastic talk. You have showed us the study for the prevalence of uveitis in the UK, and related to this question from Philippa, in those areas which are more deprived areas, what do you think is the main factor driving? Is it genetics? Is it infections?
I can hypothesize till the cows come home. I think one thing that the pandemic taught us is that there are socioeconomic drivers of inflammation. We know that, don't we? Poverty has this intrinsic link to inflammogenic procedure. So certainly genetics almost certainly has a role to play. All the big genetic epidemiology studies have not really given us that many answers with regards to genetic markers of uveitis, but almost certainly there'll be multifactorial predispositions. But if you think that it's partly genetics, but also really it's the environmentosome, it's the environmental exposome that's really driving disease, then that's when we get to the heart of it, particularly with childhood onset disease, because it's not just the environment of the child, but it's the environment of the family in particular. So the embryonic period, what's happening around the mother, the stress of the mother as well, because we know that that can have an impact on other autoimmune and also autoinflammatory disease. So the short answer is, I don't know, but the longer answer is, it's really interesting. And I think it really helps us to think about how we need to design future studies. For example, capturing adverse childhood exposures, capturing air quality metrics, hard data that we can then use with big enough data sets to really interrogate those potential drivers of disease.
Microbiota is involved. Okay, just linking to Ines. Okay, but just a last comment. Thank you for bringing a talk highlighting equity and the fact that in healthcare, we really have to think about the people where we can make the difference, and we're working in the health system. So thank you for bringing that subject too.
The other thing I should say is that TINU. So TINU went crazy during the pandemic in the UK. We found so tubular interstitial nephritis and uveitis. Your papers? Yeah, I've seen your papers. Okay, yeah, exactly. Yeah, so the incidence of that during the pandemic was much greater than we would have expected from the evidence base. Of course, part of that will be better detection, but I think we all know that there was, because it's gone down again now. So it's the challenges of this kind of work are that it's so much it's anchored within the context of the time in which the experiment takes place. So we will never be able to escape the fact that it took place during the pandemic unicorns. So the findings that we have are always interpreted with that context in mind. But it was incredibly interesting that that big push towards TINU happened during the pandemic.
Thank you. My question was pretty much overlapping Elsa's question about poverty and the likelihood of uveitis in do you think nutrition, lack of physical activity, non-compliance to treatments are all could be linked, or an explanation to this? Yes. Okay.
I think that, again, when we went into things like diet, sport, the ingredients, that's it. That's just 5 minutes a day. You don't have a good catch of what the child is eating. So, but almost
Can you comment a bit on the other 85% children with uveitis who did not have JAA? What did they have? Idiopathic mainly. Okay. Okay. That's one of our major diagnoses as well.